Hello and welcome to my channel. So in today's video we are going to discuss about peptide bonds. Since we have already discussed about the amino acids in our previous videos, so we are going to focus on the peptide bonds now. The type of amino acids which we are dealing with are L amino acids because they are the one which are synthesized inside the human body, in which NH2 group is on the left hand side and COOH group is on the right hand side. Now, whenever two monomers combine, two amino acids combine, dehydration synthesis takes place. That is, one H2O molecule is removed, OH from carboxylic acid group and H from amine group. And that's how one peptide group is formed. It's always CONH and never NHCO. That's because N remains always on the left hand side, since polypeptide synthesis takes place from N to C terminus. Now subsequently we can form larger peptides also, like oligopeptides, hexapeptide, nonapeptide. So taking an example, hexapeptide will have 7 amino acids. Now the order of polypeptides, starting from peptides to dipeptide to oligopeptide and finally to polymer of peptide, that is called polypeptide. Oligopeptides contain 3 to 10 amino acids, whereas polypeptides deal with thousands of amino acids. The first sequence, the first order of polypeptide, which is called primary sequence, the major force which holds them, that force is called force of peptide bonds. The only force present is peptide bond, so they are covalently linked to one another to dehydration reactions, forming the peptide bond and creating the long polypeptide chain. Now, amino acids covalently link to one another via C terminus of one residue and the N terminus of the adjacent residue, as we have already seen in the previous slide. Now, the ultimate shape of the protein, it is highly influenced by the primary structure, because that is the base of the building, which is the building is protein and the base is the primary structure. All these structures of proteins are derived from DNA, which is the master regulator. DNA is transcribed to mRNA, and then mRNA is translated to different codons, which finally form a long polypeptide chain. And if there is even a single mutation in any of the amino acid, it can change the entire function of a protein. The next structure we have secondary structures. The major force by which proteins are holded together in the secondary structure, that force is called hydrogen bonding. There are two types of secondary structure, alpha helices and beta pleated sheets. Now besides peptide bonds, secondary structures, they have hydrogen bonding. Alpha helices and beta pleated sheets both are held in shape by hydrogen bonds, which form between carbonyl oxygen of one amino acid and the amino hydrogen of another. So as you can see here in the structure, the carbonyl oxygen CO of 1 is forming a bond with hydrogen of NH. So now the first amino acid combines with the fourth amino acid to form this helical shape. The carbonyl oxygen of one amino acid is hydrogen bonded to the amino hydrogen of an amino acid, that is 4 down to the chain. And that's how this pattern of helical structure is formed. So this pattern of bonding pulls the polypeptide chain into a helical structure that resembles a curled ribbon, and each turn of helix contains approximately 3 to 4 amino acids, so which comes to 3.6 amino acids in a whole. Now the R groups of the amino acid, they stick outward only from the alpha helix, because that's how they are free to interact. The only exceptions are prolines and glycines. Prolines and glycerines can be a very big problem when they are present in the middle in the middle of the alpha helices, because they can either break the structure or they can form kink in the structure of alpha helices. That's why we call them helix breakers. Proline is often seen as the first residue of a helix, and it is presumed due to its structural rigidity. On the other hand, the other extreme, we have glycine. Glycine also tends to disrupt helices because of its high conformational flexibility. As we have already studied about the glycine structure, it has a very basic structure with just hydrogen as a side chain. Coming to beta pleated sheets, they can be of two types, parallel and antiparallel. Two or more segments of a polypeptide chain, they line up next to each other, forming a sheet-like structure held together by a hydrogen bond. The hydrogen bonds here, the parallel hydrogen bonds, we can see the parallel hydrogen bonding is diagonal. Oxygen of the carbonyl carbon bonds with the hydrogen of the amide group. So in this structure, when two segments of a polypeptide chain overlap with one another, they form a row of hydrogen bonds with one another. Whereas in the antiparallel arrangement, as we can see, the hydrogen bonds are straight. So the parallel and antiparallel arrangement, they are the direct consequence of the directionality of the polypeptide chain. In antiparallel arrangement, the C-terminus end of one segment is on the same side as the N-terminus end of the other segment. Whereas in parallel arrangement, the C-terminus end and the N-terminus end, they are on the same side for both segments. That's why the antiparallel arrangement is more stable than the parallel arrangement. And the pleats occur because of the alternating planes of the peptide bonds between amino acids. That's all about the video. Thanks for watching.